Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Wendt, and I'm the founder of Everything Epigenetics. Today, I am so happy to have on actually two guests with me, David Goldberg and Nicole Renke. Now, this entire episode is going to be a little bit different. We are really going to be focusing on DNA methylation tools, including Illumina's very new tool called the Methylation Screening Array, or MSA array. This is so important and why I like to cover a lot of different aspects on my podcast, because without these tools, you don't get the insights that epigenetics is able to provide us, right? We don't get the insights into disease diagnostics. We don't get the insights into biological aging or other therapeutic implications that this information can offer us. So we talk about Nicole and David and their background and how they got into this space. We're even going to give a brief overview about methylation and the methylation screening array. We're going to answer questions like, what are the applications of Illumina's microarrays and why are they used today? What's an EWAS and why are they popular for EWAS? And then why do people even like to use methylation microarrays when we have methylation sequencing tools available? We'll also understand a little bit more about the MSA, the methylation screening array broadly, and the market which needs these types of tools, including, again, researchers, diagnostic companies, even all the way down to the clinician and patient themselves. So we even talk about how this array was designed. David walks us through the content selection process, explaining all the sources of content that were used, and even talking about the new updated content this tool has on it. We then end by talking about what is the most exciting thing about the methylation screening array, and even the preprint that David and Wanding played a vital role in. Finally, we end on what other methylation products are in development and what Nicole can tell us about them. Now, a little bit of an introduction into my guests. Nicole is the global product manager of the methylation arrays at Illumina. She works with a lot of teams there to really help bring new technologies to the DNA methylation microarray business. She joined the Infineum methylation product management team in 2020 and has led a lot of these array launches. She received her Bachelor of Science and Master of Science degrees from Tulane University and holds certifications in product management, marketing, and DNA methylation analyses. As for David, he is originally from upstate New York and received a BS in biological sciences. And then he was really into researching neurogenesis in the adult mammalian brain. He's actually currently a PhD candidate at the University of Pennsylvania in the lab of Dr. Wanding Zhu, where he is developing computational methods for the analysis of DNA methylation and studying it in neurodegenerative disorders. David has worked with Dr. Zhu to develop the content on the methylation screening array, Illumina's most recent array, which we talk about today. And he's currently using this platform now to study the dynamics of DNA methylation and hydroxymethylation across diverse human tissue types. I will definitely be having both of them back on separately on the show, just because they're so interesting, and I'm personally just interested in their day-to-day work and how they're moving the epigenetics field forward. Now for my guest, David Goldberg and Nicole Renke. I hope you enjoy. Welcome to the Everything Epigenetics podcast, Nicole and David. I'm excited to have you here today. Thanks for spending your time with me.
Thanks for having us on the podcast, Hannah.
Yeah, it's great to be here. Thank you, Hannah.
Yes, I've recently been doing more podcasts with more than one person. It's super fun because we get a couple different viewpoints. Nicole and David, they each have their unique skill set. I just gave you a little bit of background about them, but I want to hear it from them too. So we're going to kind of ping pong this. I'm going to give some questions to Nicole, then go to David and keep going back and forth. Nicole, you are the global product manager for methylation arrays at Illumina. I've spoken about this topic several times on the podcast before, but from you, what does this role entail? What does it mean? What do you do on a day-to-day basis?
Yeah, so It's a really exciting role, and I'm really grateful to be in this position. Basically, what I do as a product manager is I do about 50% of my work involves kind of product strategy, and the other 50% is more on execution. So bringing new product ideas we have to the market. So as sort of the business lead of DNA methylation microarrays, I work with the development teams to bring new products to market, like I said. We also work with operations teams, marketing, and sales. So really every step of the product life cycle. And what's really important in product management is to have a really good pulse on user needs. So what I try to do is stay very close to the market, not only meeting customers in person, but also reading a lot of papers, staying up to date with the literature in DNA methylation, and bringing those insights into our product development process.
Yeah,
and again, I'm just a product manager, can't really do anything without, all these different roles that support us. So we're one piece of the pie, but I'm just so humbled really all the time to work with just brilliant scientists on the product development side, and just really great people who are on sales and marketing and actually, get out into the field and find the right sorts of users for our products.
Yeah, that's great. I have a new profound love for product teams. I never knew what that was, what that meant. I'm like, what does a product product manager do? And we now have a full product team at True Diagnostic, and it has made my life so much easier. So you're like the glue that, holds everyone together. So I love that we have you here, speaking from Illumina on kind of the creation of this new methylation screening array we'll get into. And then we also have David here. He's from University of Pennsylvania. David, what has been your research focus of your PhD, and how did you come into this world of epigenetics, specifically DNA methylation?
So I've been interested in DNA methylation for a long time now. Studied biology, and I just always found it fascinating that we have, trillions of cells in our bodies, hundreds of cell types, very different organs and organ systems, but they all have the same DNA. And it always just fascinated me how you could get such different cell types and tissue functioning from the same genome. And a lot of that is regulated by DNA methylation. So it always really interested me. Some of the early studies using mouse models where they showed that, depending on the diet that you gave the agouti mouse, it would have different fur color, different predispositions to metabolic disorders. And this was all regulated by the methylation at certain genes, which was a direct readout of the environment and the diet. So I always found it fascinating, really going from that molecular level on top of the genome to the actual phenotype. These days, during my PhD work, I've been doing a lot of research in neurodegenerative disorders. So focusing on Alzheimer's disease and primary age-related tauopathy, and trying to use DNA methylation to predict Alzheimer's or the level of tau in the brain, using just the marks on the genome, which has been really interesting journey. And then on the other side, I also work in some tool development, obviously the development of the MSA array, as well as a software program to facilitate the functional enrichment testing of CPG queries and understanding what certain cytosines are enriched in throughout the genome, to better interpret results from EWAS studies and other methylation-based analyses.
Yeah, definitely. Well, thank you, for that little bit of an introduction. I had Randy Jurtle on my podcast, and we talked about the agouti mice trial and what he's done with his work in the imprintome. So that was like a dream having him on. I remember looking at that when I was a little girl studying biology and being like, this is
Yeah.
so cool. So that's great. And then personally, I'm interested in Alzheimer's as well. I have an APOE3/4. So, being able to detect even risk from the DNA methylation level and epigenetic level, and then maybe being able to reduce that risk with said therapies, I think is a personal interest of mine too. So yeah, we're gonna hop into kind of this methylation screening array. We've said this a couple times now. That's really the main point of this episode today. You've heard me talk a little bit about kind of these microarrays or these bead chips, we call them. That's what we're physically running your sample on, at least when you send one to True Diagnostic when we measure your epigenome. So Nicole, these next set of questions are going to be, directed toward you. Can you just give us a very simplified definition of what is a methylation microarray, and then also maybe a little bit of the history behind them at Illumina?
Yeah, definitely. So in short, a microarray we can think of generally as a laboratory tool that we use to measure DNA methylation. And to kind of explain it, I think I'll go back to just the general principles of DNA sequencing. So we have, in DNA sequencing, we are basically trying to decode the genome and read the sequence of DNA nucleotides. And we have technology to do this, and it's been around for several years. The way this works is, in the simplest terms, the DNA nucleotides are fluorescently labeled, and we use a scanning device, whether it's a sequencing instrument or a microarray scanner. The function of these devices is to actually pick up on those fluorescent signals. Basically, they're colors. And what the color signal tells us is the DNA base at a given location in the genome. Now, for methylation, what we are trying to do is not to measure the DNA nucleotides or those DNA variants, but we are actually trying to distinguish between methylated cytosines and unmethylated cytosines primarily. And just to give people some context, when we talk about DNA methylation, the most common type of DNA methylation that's been the most well studied is DNA methylation, which is a chemical tag that basically attaches to the cytosine base specifically. So what we do is to measure methylation, we actually have to convert the DNA sequence to a different format that really allows us to interrogate methylated cytosines and unmethylated cytosines. But it's still based on that foundational technology where we are using these fluorescently labeled nucleotides to pick up on the methylation signal at a given location in the genome. And so in short, that's how we measure DNA methylation, is we use this foundational technology, but we do an additional step up front to convert the genome, and that allows us to figure out the methylation signal at a given location.
Yeah, that's a really great definition. And then I know these have definitely evolved over time, right? Any of the researchers listening, you go back and read research papers, and there's what I think it's a 27K array, right? A 450K array, et cetera. Do you just want to give a little bit of a history of maybe the growth and the expansion we've seen, how really far we've come with this technology?
Yeah, it's really interesting because the timeline is actually quite long, and it's been really like a privilege to work with all these researchers who have used our iterations, the very first iterations of our methylation microarrays. And when that started was actually an assay called Golden Gate, which launched in, I believe it was 2008. And this technology could target around 1,500 of these CPG sites. So these cytosines where methylation is typically known to happen in the genome. So 1,500 CPG sites is where we started. And then in 2010, we had the 27K Infinium array. So that targeted 27,000 CPG sites. And back then, I was not a researcher back then, but it must have been something that was really profound to be able to measure that many more. And another big advancement was the 450K array, which was launched in 2013, and that covers 450,000 CPG sites in the genome. So now we're up to 500,000. And this was really the tool that a lot of the field of EWAS, which is Epigenome-Wide Association Studies, was based on. So there are thousands of data sets that you can find that are publicly available on Geo that have 450K array data. And what was the next advancement was Methylation Epic in 2015. And this pretty much doubled the content of the 450K array up to around 860,000 CPG sites in the genome. And the content was improved. So we were learning about, what were the important areas to cover in the methylome. And this content really expanded the coverage into not only the promoter regions, which were very well established at that time, but also getting into new areas like enhancers and intergenic regions. And then in the last few years, we've also expanded the product portfolio beyond humans, and we launched a mouse methylation bead chip in 2010. So that is very useful for researchers who are studying model organisms, and that is around 285,000 CPG sites and can sort of be considered an epic for mice. Mm-hmm. And in late 2022, we came out with Epic version 2, which has some expanded content that covers more enhancers, super enhancers, CTCF binding sites. And like you mentioned, Hannah, what we're really excited about these days is our new methylation screening array. And this, I really had the pleasure of working with David and Wan Ding from the Children's Hospital of Philadelphia. And this tool is really designed to be the ultimate EWAS tool that I believe is going to take us into the next generation of epigenetics research and really lead us into applying epigenetics in the population health context to very large screening cohorts. So for MSA, we were really focused on building a tool that was a lot more scalable, a lot more affordable, and had the right content selection because this is a targeted tool. So our priority with the content was kind of twofold. We wanted to enable researchers to be able to use the same powerful CPG sites that have been discovered to be associated with a wide variety of traits and cell types from the whole history of EWAS and all of the literature that's been published to date. And we also wanted to enable new capabilities, so new biomarkers that have been so new methylation sites that could be very useful for research. And this is based on, new research, cell type atlases, new sort of whole genome bisulfite sequencing data sets, and databases that have been built up since EPIC was launched in 2015. So to kind of execute this vision, we had to outsource because this is something that we knew we needed some bioinformatics expertise on, and this is where Wanding and David came in. So Wanding and David have Wanding's lab has been very instrumental in the previous designs of Infinium B chips, like the mouse methylation array. And so we reached out to them, and they were really the content leads to help bring this vision we had to life. And we're just so grateful to be able to work with them, and just really appreciate their collaboration ability. So not only did they kind of look through all the literature, but they also reached out to other users, and we're just very collaborative.
Yeah, thanks for that history, Nicole. That's awesome. And remember, you all listening, like, this is the tool Nicole and David mentioned at the beginning to measure DNA methylation. So without this tool, we can't have all of this awesome science and research coming out of this. So, there are a ton of clinical implications of DNA methylation. We know, DNA methylation patterns are altered in all types of different disorders. David mentioned he's studying more like neurodegenerative, neurodevelopmental maybe disorders. There's, episignatures as potential biomarkers. So being able to even report out other like clinical lab values or other parts of the multiome through DNA methylation, as well as other clinical implications and further research and therapeutic implications. They're obviously huge in the epigenetic aging clock space, which I talk frequently about, even all the way to environmental exposures, methylation risk scores that I spoke with Michael Thompson out of UCLA on, and then what Nicole just mentioned too, right? These EWAS studies. So the methylation array, that the screening array, the MSA array we're talking about today, I think can really help EWAS, and we'll talk about how here in a second. But remember, those EWAS are just a really a type of research that measures populational level differences in DNA methylation on a large genome wide scale, but it's going to be methylome wide, right? Because we know DNA methylation is really the most stable epigenetic marker. And technology for measuring other epigenetic markers hasn't really been robust historically. So I wanted to give, again, a wider viewpoint on why it's so important to talk about this new tool and this new technology. Now, David, this one's going to be for you. What is your perspective of just methylation microarrays in general as a user of them? Like, why do people still use these microarrays? Why is Illumina coming out with all of these new versions of them when there is methylation sequencing even available? So kind of a two-parter, how you use them as a researcher, but why do we even use them still over the sequencing too?
Yeah, so there's a lot of advantages to using the microarrays. First and foremost, as Nicole mentioned, is the scalability. For whenever you want to study a trait in the population, whatever type of disease or environmental exposure, you typically want to profile a lot of samples, especially because humans have a lot of inherent variability. Different ages and sex, things like that could impact your DNA methylation. So if you really want to focus on one trait, you need to have a very large cohort. In doing whole genome bisulfite sequencing at this time, it's just not economically feasible. So the arrays are great to really do these population scale studies. And MSA is really going to be pushing that to even the next level in terms of the volume of samples that could be profiled. Another major advantage is the data is very accessible to work with. So there's a lot of tools, because this has been the main way of profiling methylation in populations. There are a lot of software packages out there that have already been designed and tested by the community for analyzing the array data. And a lot of models have been developed as well. Like you mentioned, many of these epigenetic clocks are very well characterized and in use now. So if you have a new data set or you're studying a different disease, you can apply these existing models and see if there's any sort of acceleration in what that model is reading. I think another important advantage is the quantitative resolution. So when you do sequencing, oftentimes you'll get parts of the genome, but at any given CPG site, it's very rare that you'll have very high coverage, meaning you'll have very few reads that actually have mapped to that individual CPG. Whereas on the array, some people have compared it to like 50 to 100x coverage. Your measurements have very high precision, and you can be very confident most of the time with the methylation readout that you have at those sites. And for those reasons, you don't have to deal with a lot of the data sparsity challenges that you have with whole genome bisulfite sequencing data, where you often have to merge the methylation over a genomic interval, and you start getting a lot more sort of coarse measurements. And yeah, I mean, less computing power is required, less memory, and just really well-developed tools and pipelines. So it's a pleasure to work with, and you can really profile a lot of samples. So I'm a big fan, and it's been good.
Good, good. No, it sounds like it. I mean, it just sounds like there's, many more advantages, or it makes sense that we're looking or still using these microarrays for specific use cases, I guess, depending on your area of research interest. So just to summarize too, David, you said like scalability, cost and volume considerations, accessibility to the data. So it makes so much sense. There's all these software packages. There are all these tools that you can already use that have been validated within the community. Quantitative resolution, you don't have to worry about, not having a good enough or a good amount of coverage. The microarrays are really great at that. And then, yeah, less computing, right? Who wants to do more computing these days? So it has, less memory, easier data to work with as well. So thank you for that. Now, what we've been hinting at this whole time and what we really want to talk about, this new methylation screening array. Nicole, Illumina has launched it this year. Tell us more about it. I know why we're so excited to be able to use it at True Diagnostic. So I'll definitely add my perspective, but we want to hear it from you first. So yeah, walk us through the market. Like, why did we need this? Why was this a product that came to the table and became available?
Yeah, I'll actually start by maybe adding on to, the previous points about microarrays and their advantages. So what we see today as an advantage of microarrays and why they are still around in the context of studying methylation is that they're very reproducible and quantitative, and they're able to call methylation differences in a way that they're able to call methylation differences in the context of EWAS that can lend themselves well to a very powerful, very reproducible biomarker. So there's been just thousands of papers that have been published on using DNA methylation microarrays to discover biomarkers that are associated with something like cancer or even environmental exposures like arsenic exposure, air pollution, a whole array of diseases, as well as just natural phenomena like aging. And so there is, of course, a role for DNA methylation sequencing, and those tools are going to become better and better. Illumina is also investing in new technology platforms for DNA methylation sequencing. And I think where this is really useful is to discover sort of new areas of the methylome that haven't been discovered before, kind of on a genome-wide level, and understanding really at a deeper level some of the biological pathways and how genetic variants interact with these whole regions of methylation differences that we see between cases and controls. But for methylation microarrays, they've really been a popular tool. And over the past few years, we have studied how people are using these tools. And what we found is that users really, we really have two different groups of users. So one of the groups, group one, is cancer researchers and also researchers that are studying, researchers who are studying rare genetic disorders. And what's interesting about these types of diseases and disorders is that the methylation differences that you see between, on average, your cases and your controls are actually very large. So oftentimes we see 20% or more methylation differences between, say, the methylation patterns of let's say, a tumor sample. Say you're studying lung cancer and you take a biopsy of the lung tissue and look at the DNA methylation patterns across a whole variety, across a number of people who have lung cancer, and you compare those patterns with, a biopsy of normal lung tissue, you actually are able to detect methylation differences that with a high amount of power in a small number of individuals. So we actually only need small study sizes in certain research areas. But the vast majority of our researchers who are using methylation microarrays are using them to study common diseases. So like I mentioned, things like psychiatric disorders, public health, aging, environmental epidemiology. So, exposures of, say, maternal smoking, things like that. So what the difference between these users is they actually need to have a higher number of samples to make those methylation biomarker discoveries. And that's because the methylation differences we see in common diseases or environmental exposures are actually quite small, often under 5%. And in order to find these robust biomarkers that you could, say, use to develop a classifier, you really need a lot of samples. And this is what we refer to these users as kind of the EWAS research type. And over time, we've heard that these types of researchers, they're actually looking to run studies of at least 1,000 samples. And when they're studying diseases or exposures with even smaller predicted methylation differences, they actually need to scale that research to 10,000 samples or even more. And at the same time, we have a lot of national genomics initiatives that are popping up and have already done sequencing or genotyping of an entire cohort and are using these insights to improve population health. And the field of epigenetics, it's really becoming very, it's gaining a lot of traction. People now are interested in studying epigenetics, and DNA methylation is the most available type of biomarker to study within epigenetics. So we are seeing these big projects that have a need for methylation profiling. So kind of a long explanation, but the methylation screening array was a tool that we knew that we needed to be more affordable than the existing tools we have. We knew that it needed to be more scalable. We needed a tool that would be able to profile, say, up to 1 million samples in just a couple of years. And we needed a tool that had content that was going to be very valuable for EWAS researchers and for the study of diseases that would be considered under the umbrella of population health.
Yeah. Yeah, no, I know that's comprehensive, and you said, it's a longer answer, but I think it's really good to touch on those points. So, from a laboratory standpoint at True Diagnostic, I mean, this is more affordable. Than we can pass off that cost to, our end consumer, and start to make these diagnostic based tools much cheaper to the end user. I think that's really the goal, is being able to get these into everyone's hands, not just people who can actually afford it. So by you all doing that, it helps us. It helps, the end user, the public, as well. It's super scalable, as you mentioned. So, we used to only be able to run 8 samples on one of these microarrays, and now we can run 48, which is amazing. So it allows us to have a much more higher throughput in our laboratory. And then you mentioned the content too. We have defined content in terms of CPG locations and particular areas of the methylome that we want to look at. So being able to have that defined content and maybe a more concise microarray, it just makes our lives a lot easier as well. And that's the next point I really want to go to, is that content piece. David, I know you played a massive role in the content design of the methylation screening array, the MSA. So I'd love to hear a little bit more from you, how you and Juan Ding actually chose the content. So feel free to walk us through the selection process, all of the sources that were used. I get this question a lot, like, who comes up with this stuff, right? Like, who picks what we're actually looking at? And I have him here on the podcast, so you all can hear from him directly.
Yeah, so well, fortunately, a lot of people really helped with the content selection, because we really leveraged the thousands of EWAS studies that were done, and a lot of the prior probes that were from previous platforms. So basically, the array was designed in 2 phases. The first half was trying to identify previous probes from Infinium platforms um, that had a lot of associations, were statistically robust, and associated with a diversity of traits. So essentially what we did was we curated thousands of studies from EWAS databases, a lot of manual curation, talking to people in the fields that are doing population scale EWAS studies. And what we did was group them into approximately 20 different trait categories. So we were able to use some natural language processing to group them into different traits. And these range from cardiovascular to environmental exposures, to genetic and psychiatric, many different traits. And then within those different trait groups, we were able to find studies. We were able to basically scrape the statistics on those studies, to look at the cohort sizes, the impact factor of the studies, and then rank the probes according to their statistical significance. So we were able to prioritize the studies and get a broad diversity of trait groups, and prioritize the probe selection that way. And then from all of our other studies, we were able to just downsample and select a small number from what we deemed as the lesser statistically robust studies, but hopefully still capture some markers from the traits that were examined in those studies. So we had roughly 20 major trait groups with thousands of studies and thousands of subtraits, which we think encapsulates a lot of different biology. The next phase of the design was, in recent years, there's been a lot of large-scale whole genome bisulfite sequencing projects. So we wanted to sort of combine these advantages we discussed for the microarrays, where you could have a select CPG site with high quantitative precision, with the knowledge that's been gleaned from these WGBS studies. So we looked at thousands of different single cell and sorted WGBS data sets, and we had different target biology groups that we wanted to find markers for. So we were particularly interested in tissue-specific and cell-specific CPG sites. So these are markers in the genome, CPGs, where the methylation or the absence of methylation essentially defines the cell type and distinguishes it from all the other cell types that might be in the tissue or in the body. So we were very interested in those, and we did a lot of analyses over thousands of methylomes to find markers that distinguish hundreds of different cell types. And I'll talk a little bit more later on about some of the applications of that. We were also interested in sites that where the methylation might actually be correlated with gene expression. So in these studies, you do an EWAS. If you identify a site that associates with a trait, you ultimately want to understand what it might be doing or what the biology is behind that methylation trait association. And usually, if a methylated site is going to have an impact on a phenotype, it's probably going to be impacting gene expression or genome stability in one way or another. So we were able to leverage these matched WGBS RNA-seq data sets to find the CPG sites where the methylation actually correlates with the gene expression. So that's another example of one of the design groups that we targeted. Then we had about 15 more other biology groups that we were able to mine WGBS and other sources for. But altogether, there's a lot of new biology. A lot of the new probe sites target cis regulatory elements. So these are functional elements in the genome that are predicted to have a role in gene expression and regulation of the genome. So there's about over 100,000 new probe sites that target these different biologies that might have a lot of discovery power and might lead to novel associations. And not just for methylation, but hydroxymethylation as well, which is a related epigenetic mark. So yeah, it was leveraging both prior EWAS studies as well as new whole genome bisulfite sequencing data where we were able to identify a lot of these markers.
Awesome. No, it seems what I was saying, David, like it's super comprehensive, right? So I'm excited to learn a little bit more about how you all were able to pick the probes and the content design of that MSA array. Now, just very briefly as well, if you were to pick like because there's so many, let's confine it to like one or two areas or pieces of content. What do you think would be most useful for those performing those EWAS studies? Does anything stick out in particular?
Yeah, well, it's hard. I think they're all going to be pretty useful. Maybe I'm a little biased, but I really am excited about the cell-specific methylation patterns that we were able to identify and validate in some of our initial studies. I think that most diseases and traits ultimately manifest as altered cell type proportions or altered functioning of the tissue type where the trait manifests. And these markers will really enable high resolution deconvolution, or I think they'll enable the study of more rare cell types in heterogeneous bulk samples. So normally when you do an EWAS, you take a biopsy of tissue, but that tissue has many different cell types. And if the one that's implicated in the trait is relatively rare, then you need more markers for that cell type. But I think as a result of this cell-specific methylation, it'll give the ability to really deconvolve bulk tissues and understand how different traits are altering the more rare subtypes. And to that point, previous EWAS studies have tried and have done deconvolution, but it's on a more coarse level. So for example, in the brain, I do some work on neurodegeneration. Oftentimes the deconvolution will only be looking at neurons and non-neurons. But now, as a result of these single cell WGBS studies that have been done in the brain in the last couple of years, we know that there are hundreds of cell types. And now I think we can actually develop references in deconvolution panels to better estimate the proportions of more rare cell types in EWAS studies, both to covary for cell types, but as well to identify the populations that might be vulnerable in a given exposure or disease. So I think that'll be one of the major strengths of this new platform.
Yeah, no, that one sounds exciting for sure. Well, we're getting to the end of this podcast. I have kind of one more question for each of you both. Nicole, I don't mean to leave you hanging, but I'm going to save yours for the end. So I'm going to bombard David with one last question here before we start to close this out. So just kind of, again, adding on to the topic we're already mentioning regarding, how we created the MSA, the content that you put on it, what you think would be most useful for researchers, especially those with EWAS, kind of the single cell profiling. You and Wanding were really heavily involved in just the overall creation of this MSA bead chip. I think that's super clear. You published a preprint as well. Maybe by the time this is published, it'll be pushed to publication. Regardless, we'll link it in the show notes. But I know a big concern of not only users of the microarrays, but even down to whoever's receiving the end result from the microarray, whether it be a diagnostic tool or a report, is always wanting to know about the technical performance and the biological validation, which, it was run on. Can you just talk very briefly about that, David?
Yeah, so basically we go through a series of sort of technical experiments to check the performance. So we'll generate DNA and methylation profiles for cell lines, and we'll look at the cross-replicate correlations. And in those experiments, the MSA readings between technical replicates were highly correlated and performed at a similar level to previous platforms. We also compared the accuracy, comparing it to other sequencing technologies, and found extremely high correlations with both WGBS sequencing as well as some other capture sequencing approaches. So the performance is as good as the previous platforms in our experiments, which we were really happy and relieved to see. In terms of the biological variation or validation, we've now profiled, so the original preprint doesn't have all of our data, but our revisions will. We've profiled hundreds of tissues, both for methylation and hydroxymethylation, and are across dozens of tissue types. And we're seeing very nice capacity to do deconvolution of the tissues. When we do unsupervised clustering, we're able to see that the array is, the methylation profiles are clearly clustering the tissue types. And we validated a lot of our markers, which is great. And then we've also tested the epigenetic clocks that we included in the design, some of the major ones that are used in the field. And we see very high correlations between the predicted epigenetic ages as well as with the chronological ages of our samples. And there's a lot more biology that will be in our paper, but so far the technical performance looks great, and we're learning a lot of interesting biology from the samples, the actual tissue samples that we profiled. So
Great. Yeah, I'm excited to read more about the biology involved in that validation once we get those edits to the preprint in as well and it's published. So sounds great. Back to you, Nicole. Sorry to leave you hanging there for a second. Last question for you. What excites you most about the methylation screening array? Like, why do people have to be using this?
Yeah, so I'm really excited about the methylation screening array because we're really fulfilling a very large user need. And like I mentioned before, I think this tool will enable the next generation of methylation research. So really the application of methylation profiling in very large screening projects. And we might even see this at the government initiatives level. For example, Million Veterans Program. There's a lot of new government initiatives and biobank projects that are seeking to be able to better predict diseases and to better predict how to treat diseases and prevent diseases. So I think getting methylation, all of the methylation insights that have been found, we in the epigenetics community know how powerful DNA methylation is as a biomarker for human disease, but getting those insights to scale and actually impact human health in a very large way is really exciting to me. And I'm just, I have kind of what I enjoy most about my job is just seeing all the applications that have been developed and continue to be developed. So I think we're going to see more aging clocks and more methylation risk scores and new classifiers in all sorts of common disease areas that will be enabled by this methylation screening array.
Yeah, definitely. I have to sneak one last question in for you, Nicole, too, because I'm just personally selfishly interested. What's next in the methylation products in terms of development? So just very quickly, is there anything you all are looking at in the future? I know you mentioned, maybe reevaluating or relooking at the whole genome bisulfate sequencing and maybe, adding better tools for more of that discovery-based purpose. But like, is there anything on the pipeline? What can you tell us about that?
Yeah, definitely. I'll say broadly, Illumina is investing in DNA methylation technologies, whether it's microarrays or sequencing based approaches. And first, to just dispel the myths, I think, that have been present for over a decade now is, people have been forecasting the death of microarrays. And what we've seen in the methylation microarray business is that, consistent growth over year. And as a result, Illumina is actually investing more into methylation microarrays than they ever have been. And I can't share details of specific products, but what I can say, yes, we are interested in different types of chemistries that are emerging, and but really our goal is to just amplify a lot of the benefits that bring people to the platform. So like we've mentioned, reproducibility, scalability, affordability. We're working on all of those things. And then we're also going to be focusing a lot on the user experience. And like David said, there's a great set of existing tools, but how can we make it easier for, say, new researchers to get from data to biological insights faster? And so, I can promise you that at the forefront of all our product developments will be really trying to understand what our users need in terms of new content. And my most important priority is staying really close to the user community. I always try to make myself available, so you can reach out to me if you have any product ideas or ideas to improve, say, the content of our products. I can be reached on X at Nicole Renke or on LinkedIn, also Nicole Renke. And I'm always really interested in connecting with our users.
Yeah, I love that. That's awesome. Yeah, the microarray, it's not going anywhere. It's here for a very, very long time. Thanks to both of you. Nicole, you just gave your information in terms of, where listeners can come to find you. David, what about yourself? If anyone wants to reach out, I'm sure you have your UPenn profile and whatnot. But how can people get a hold of you if they're interested or look at some of your work?
Yeah, my email in the lab. So I'm in the lab of Dr. Wanding Joe, which hopefully we can drop a link to the lab website. We're doing some really cool stuff with DNA methylation and developing informatics techniques. So I can be reached through there as well. And hopefully, hopefully someone reaches out.
Of course. All right. My fun question at the end of the podcast. I was itching to get to this one. I don't care who goes first. It's, if you could be any animal in the world, what would you be and why? You have to give an answer.
Okay. Well, for me, I think I would be a sea turtle because I think they
Ooh,
just move so gracefully through the water and they just look so calm. I think they're just a beautiful creature.
love that. I don't think I've ever gotten that answer before, so that's a good one. David, what are you thinking?
Well, I definitely don't want to be a mouse. I don't want my DNA. I don't want my methylation getting. But yeah, maybe a bird of some sort.
Okay.
Have the ability to fly and go long distances, see places. So we'll go with an eagle, maybe.
There we go. That's a good one. Well, no, this has been great, Nicole and David. Thank you so, so, so much. So excited to learn about the new methylation screening array and the history behind the microarrays within Illumina. Thanks everyone else for listening and joining us at the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more about how. Thank you all.
Thank you, Hannah. Take
Thank
care.
you.